Let's begin with a simple but important reality. In pharmaceuticals and medical devices, patient risk doesn't only come from the drug substance. It can come from the materials surrounding it: packaging, container closures, tubing, or device components. Extractables and leachables, often called E and L, address this exact risk. Regulatory agencies like the FDA expect manufacturers to understand how materials interact with products over time and how that interaction may affect patients. This presentation walks through the FDA-aligned framework for E and L studies, showing how science, toxicology, and risk management come together to ensure patient safety and regulatory success. Extractables and leachables are chemical compounds that can migrate from packaging systems, container closures, or medical devices into drug products, or directly into the human body. These chemicals are not formulation impurities. They originate from polymers, elastomers, adhesives, inks, lubricants, or even manufacturing residues. The challenge is that many of these compounds are invisible to the naked eye. Yet even very small amounts can matter, especially for injectables, inhalation products, or long-term therapies. E and L testing is driven by clear regulatory expectations. The FDA requires E and L studies to prevent adverse health effects and expects reliable, well-justified analytical data. ICH Q3E introduces a structured, risk-based framework for pharmaceutical products and combination products. USP chapters such as 665, 1663, 1664 and 1665 provide detailed technical guidance on polymers, extractables, leachables, and risk assessment. For medical devices, ISO 10993-18 is the globally accepted standard for chemical characterization and is recognized by the FDA as a consensus standard. A risk-based approach is the cornerstone of E and L evaluation. The goal is not to test everything blindly, but to focus on what truly impacts patient safety. The process begins with risk identification, using prior knowledge, supplier data, material composition, and extractables testing. Next comes risk analysis, where potential leachables are quantified and patient exposure is estimated. Risk evaluation determines whether the exposure is acceptable, and risk control implements actions such as material changes, pre-washing, or additional purification. ICHQ3E strongly emphasises that material selection must be driven by quality risk assessment, not convenience. E and L studies must be carefully designed to be both scientifically sound and regulator-ready. Study planning defines the scope, materials, solvents, and endpoints. Extractable studies use aggressive laboratory conditions and focus on identifying potential compounds. Leachable studies simulate real-world storage and use conditions, include multiple time points, and directly assess patient exposure. When studies are designed correctly from the start, they prevent regulatory delays and reduce the need for rework. Sensitive and validated analytical techniques are essential for E and L studies. LC-MS/MS is used for non-volatile and semi-volatile organic compounds. GC-MS targets volatile extractables, while ICP-MS is critical for elemental impurities. A key concept here is the Analytical Evaluation Threshold, or AET. The AET defines which compounds must be identified and reported based on dosing and patient exposure. This ensures analytical effort is focused on compounds that matter from a safety perspective. Analytical detection alone is not enough. Identified leachables must be evaluated using toxicological thresholds. The analytical evaluation threshold determines reporting. The threshold of toxicological concern, or TTC, addresses systemic and mutagenic risks. The safety concern threshold, or SCT, applies to non-mutagenic toxicity. Exposure duration and route of administration are critical. What is safe for short-term exposure may not be acceptable for chronic use. This step translates analytical data into meaningful patient safety decisions. For medical devices, ISO 10993-18 provides a structured framework from material identification and chemical composition to extractables estimation and leachables measurement under clinical conditions. An important regulatory advantage is that chemical characterization can sometimes replace biological testing for certain endpoints. Equally important is lifecycle management. Any change in formulation, packaging, supplier, manufacturing process, or dosing can alter the E and L profile and must be reassessed. Compliance is ongoing, not one-time. For regulatory submissions, comprehensive and well-structured documentation is essential to demonstrate extractables and leachables compliance. This includes clear study design justification, complete E and L study reports, validated analytical methods, documented AET listings, and a formal safety risk assessment aligned with ICH-Q3E. Equally important is lifecycle management. Any changes to formulation, container systems, manufacturing processes, patient exposure, or product indication require re-evaluation to ensure continued patient safety and regulatory approval. Successful E&L programs start early and follow a structured strategy. First, define the study scope. Then, design risk-based extractables and leachables studies. Validate analytical methods and document everything thoroughly. Early implementation avoids regulatory setbacks. Lifecycle management ensures long-term compliance. And expert partnerships help accelerate approvals. Ultimately, extractables and leachables studies protect patients, strengthen regulatory confidence, and support sustainable product success. Thank you for your attention.